The title of this presentation is Genetic Modification of Mice Without the Need for Extensive Breeding. And the aim of this project is to reduce the number of mice required to perform research with genetically altered mice. Development of methods to produce gene alteration in mice has caused a biomedical revolution and has provided a lot of benefits. The gene alteration has, for example, enabled very close modeling of human diseases, ensuring that the data generated by the animal models can be easily transferred into a human setting. And moreover, this method has allowed for a more accurate investigation of gene function in that we do not have to rely 100% on pharmacological inhibitors, for example, which can have side effects. However, these conventional methods for producing the gene-altered mice has also caused a very dramatic increase in the number of mice that are needed to perform animal experiments. Why are so many mice needed? The conventional methods of producing gene-altered mice relies on the modification of their genomes. Therefore, a significant fraction of the mice are only used for generation and maintenance of a stock. And it has been estimated that only 25% of the animals are used directly for experiments. Therefore, our aim was to develop an alternative methods where we can induce the genetic modification in the mice without extensive breeding, and we can thereby eliminate a significant fraction of the mice needed to perform the experiments. One method to make gene alteration in mice is via adeno-associated viruses, or AAVs. The AAVs are safe and very straightforward to produce in the lab, and they are simple. So the AAV particle, they consist of only two components. Their genetic material and then a protein coat or a cap that surrounds and protects this DNA string. So once these particles are injected into, for example, a mouse, the coat proteins here, they interact with specific receptors on cells, and then the particles here are internalized and can deliver the DNA string to the cell. However, there only exists a very limited number of AAV coat proteins. So only a very limited number of cells can be targeted in vivo. We therefore wanted to expand the number of code proteins of the AAVs and thereby increase the number of cell types that can be targeted in vivo. To do this, we relied on a method described in 2016, which allows for selection of AAV variants that cause transduction of specific cell types. And in this method here, a library of the gene encoding for this code protein, also called CAP, is produced in the laboratory. And then AAV particles encoding the genes for this CAP protein here are produced, and the results is a unique AAV particle with a unique CAP protein here, which contains the DNA string containing the specific CAP gene. The library of AAVs is then injected into a mouse where the cells that needs to be targeted are expressing a molecular scissor called CRE. Thus, when these AAVs enter the cells, the DNA string is released, and if the cell expresses CRE, this will cause a molecular rearrangement of the gene, enabling us to PCR amplify the CAP gene. We can then DNA sequence this CAP gene afterwards, and we can then identify the specific code protein that allows for transduction of these cells. We thereby identify new CAP genes that target specific cells and expand the use of this AAV approach. And to set up this technique, we use this method here to identify novel AAV variants that target the kidney, the vascular smooth muscle cells, and also the fat cells localized around arteries. We identified some new variants of the CAP genes, and to test the performance of the AAV particles that are produced with these CAP genes, we made AAV particles in the laboratory which contained a gene encoding green fluorescent protein. And we then injected these particles into mice. And the idea is that when these particles enter a cell, these cells will be green, and we can then easily identify which cell types are transduced by this new AAV particle. The project is still running and the data here are still preliminary. However, we have identified new AAVs that target kidney cells, as shown here, where a specific epithelial cell in the kidney expresses a green fluorescent protein. Moreover, we have found that AAVs can target both kidney cells and also adipocytes or fat cells that surrounds the vasculature, and also the vascular smooth muscle cells in arteries. We are currently repeating these localization experiments and optimizing the dose needed for optimal targeting of the AAVs to these specific cell types. We therefore believe that the AAV-based approach provides several benefits compared to the conventional method of gene alteration in mice. First of all, the AAV-based approach here is considerably faster than the conventional method of gene alteration and only takes around 1.5 months to create a new mouse model. Secondly, both the cost and labor involved in generating these mouse models here are significantly reduced compared to the conventional method. And it's also fairly easy to generate these AAVs, so most molecular laboratories have both the techniques and the expertise present to do this, and so these AAV-based approaches here will be fairly easy to implement in a new lab. Finally, the AAV-based approach here allows for 100% of the mice to be used to generate experimental data, and it will therefore significantly reduce the number of mice needed to do research. Finally, I would like to thank my collaborators on this project, which is postdoc Lasse Steffensen and lab technician Natasha Stanich. And I also would like to thank the Danish 3R Center for financial support for this project.